Okay, welcome to attempt number 2 to record this video. I was about halfway through, and then I got an error message from Snagit that said, could not record video, error, blah, 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 ref, blah, blah, blah. And then it didn't record, so that was a huge waste of my time. But let's try again. Okay, so what we have here is a helpful 3D model of the sectional anatomy of the spinal cord in situ. So in situ means in the place it should be ordinarily. So in this case, because the spinal cord is housed inside of the vertebral foramen of our vertebrae, in situ here means this model is showing you where it's supposed to be. It's not been imaginarily plucked out and represented somewhere else. So ordinarily, I get rid of the annotations on these videos, but this one has them embedded, so I can't do anything about that, but that's okay. It's helpful to have them sometimes. So some things to note before we start. Very obviously, this is a cervical vertebra, so I can tell because the spinous process is bifid spinous process, and I can also see here is the transverse foramen, and inside of it we've got vertebral artery. So, like I tell my students in my class, if you look at a vertebra and you see 3 holes, not one, and you see a spinous process that's forked like a snake's tongue, automatically it's cervical. So, let's just go ahead and zoom on in and start seeing what there is to see. Okay, so one of the first things that you're going to notice is the gray matter here, which is neurosomata, so cell bodies of neurons, and some interneurons and unmyelinated axons and glial cells, is shaped like a butterfly. And the top of the wings of the butterfly touch the edge. The bottom does not, and that's a really easy way to orient yourself and figure out which side of the vertebra and, excuse me, not vertebra, which side of the spinal cord is which, because the dorsal gray horn touches the edge of the spinal cord segment, and the ventral gray horn doesn't. So even if you don't have a vertebra, so your spinal cord segment is ex situ, not in situ, you can still tell dorsal from ventral because of the gray horns. There's also a lateral gray horn. So that's some useful anatomy for you. So with regard to dorsal and ventral, of course, directional terms matter in general because we are striving always for clear and unambiguous medical communication. But there's another reason that it matters for the spinal cord, and that is because the spinal cord is divided spatially into incoming afferent information and outgoing efferent information. They're not mixed together. So for my students, I used the following acronym. Oops, I started with the wrong one. So that acronym is SAME Dave. So this is a way to keep track of the meaning of the terms afferent and efferent and how that applies to the spinal cord. So SAME is sensory equals afferent, and motor equals afferent. And then Dave applies this principle to the spinal cord. So I'm going to have to move this over a little bit. Actually, I'll just erase this. If you want to rewind, you can, because this is going to be on the internet. So applying to the spinal cord, the dorsal side of the spinal cord contains incoming afferent or sensory information, And the ventral side contains outgoing information or efferent information. So because afferent and efferent basically differ only by one letter, and that's confusing to students, you can help keep them straight using this same DAVE acronym and then apply that to the spinal cord. You can also note that afferent starts with an A, which is corresponding to arriving information to the CNS, and efferent starts with E, which corresponds to exiting from CNS. So, why does that matter here? Well, dorsal and ventral in the spinal cord are separate from each other and contain separate directionality for information. So I'm going to make myself a little key here. We're going to have green be afferent / sensory, and pink be afferent / motor. So this is the dorsal root, and it spreads out into dorsal rootlets. You can kind of see here it spreads out, but from this view you can only just see one of the rootlets, the one that's closest to you. So this dorsal root contains only axons of sensory neurons. Oops, and I wrote it in the wrong color. Look at that. Let me try that again. Okay, so axons of sensory neurons. And the ventral root and rootlets, I said pink. There we go. The ventral root and rootlets carry axons of motor neurons. So efferent. So the direction of information and the direction of the action potentials that are traveling is that way. And up here, the direction of information and the direction of action potential propagation is this way. So what that means is incoming sensory information first arrives at the dorsal gray horn, and any outgoing motor information initiates from the ventral gray horn. And we'll round out some more information on that when I zoom in a little bit and make a map of which populations of cell bodies are arranged in which places. So if we look over here, what you're going to notice is that if we follow the dorsal root, it has this big bulge in it, and the ventral root has no such bulge. So the bulge has a name, and that name is the dorsal root ganglion. So I'm going to erase some of the labels as I go so I have more room to draw. So one thing you could potentially do is take screenshots of the drawing as it develops and changes so you don't miss anything. So this thing, well, let's make it green. It's the dorsal root ganglion. So it's worth taking the time to define ganglion. So there's different terminology for the arrangements of cell bodies and axons in the central versus the peripheral nervous system. It's also important to remember that the brain and spinal cord are the CNS. Anything that's outside of the brain or spinal cord is peripheral nervous system. So a ganglion is a group of cell bodies, neural cell bodies. So I'm going to write oops, I need to move this, somata. So soma means body, somata means bodies, and neuro means neuron. A group of neurosomata in the peripheral nervous system. Groups of cell bodies in the central nervous system we call other stuff. So Why is there a dorsal root ganglion, a group of cell bodies, and there's not one for the ventral root? Well, one thing that you might not have learned yet, or you might have forgotten, is that there are structural types for neurons that are based on their shape. And one of those structural types is this one, which is called a unipolar neuron, where I'll label it for you. There's some dendrites at one end, and then the initial segment of the axon is not emanating from the cell body, it's just right after the dendrites. And then this is the axon, and the cell body sits off to the side of the axon on a little stalk. So this shape name for this is unipolar. So unipolar neurons are overwhelmingly sensory neurons. And so if you have a bunch of sensory axons traveling together, it makes sense to put all of their cell bodies in the same place. So if I was going to draw our little incoming sensory neurons, you have to put their little neurosomata somewhere, right? And so they all hang out together in the dorsal root ganglion. And they're accompanied by glial cells called satellite cells, which help them deal with neurotransmitter stuff and all kinds of other things. So the ganglion, because it's housing cell bodies and glial cells, creates a bulge, which is visible. So there's a dorsal root ganglion on the dorsal side, and there's one on the left and one on the right. So these neurons and axons are going to come into the posterior gray horn, which is why it touches the edge. And depending on what kind of information it's delivering, it will synapse on some cell bodies in those horns that I will map out in a little bit. But because the cell bodies are out here and not in here, there's a dorsal root ganglion. Now, for motor stuff, the cell bodies of motor neurons are ensconced firmly in the ventral gray horn. So motor neurons are also mostly multipolar, so it's a different structural type as well. But the cell body lives in here, and then it projects its axon out through the ventral root. And so because the cell body is here, there isn't a ventral root ganglion because there doesn't need to be. The location of the cell body is different. So that's the reason there is a dorsal root ganglion, but not a ventral root ganglion. Okay, so now I'm going to erase all of this, and we're going to zoom in, and we're going to look at the gray matter in particular. And I just really hope this doesn't cancel my video recording. Okay. I just want like an intermediate amount of zooming. Is that too much to ask? Okay, perfect. All right, so you can see this is mislabeled. I'm going to have to contact the model creator about that. So notice it says anterior gray horn here and posterior here. Wrong. Okay, the rest of this I like though. So, some stuff to note here. There's a little hole that runs down the middle of this, and that is called the central canal of the spinal cord. It's full of cerebrospinal fluid. The little chunk of gray matter that is just in front of it is called the anterior gray commissure. And then in front of that, there's a little bit of white matter that also goes across, and that's called the anterior white commissure. So again, a commissure is a place where decussation is possible, so the crossing of information from the right to the left or the left to the right. Okay, so I mentioned I would give you a little map here, and we're going to just fastidiously ignore these incorrect labels because I can't believe they made that mistake. Shame, shame. Okay, so we already established that sensory is in green, so I'm going to use shades of green because we also have to worry about visceral sensory versus somatic sensory. So when those axons coming in from the periphery arrive and they synapse, they're going to synapse on cell bodies in the dorsal gray horn at one of two locations, either the top, which are cell bodies of somatic sensory neurons, or a little further down, cell bodies of visceral sensory neurons. So somatic senses are your basic body senses, so touch, proprioception, detecting of where your limbs are and what your body position is, skin sensations, pain, etc. And then visceral sensory neurons are going to be a mixture, but this is senses from your viscera. So that includes things that rise to your conscious attention, like pain and whatnot, like if I have a tummy ache, for example, but it also includes visceral sensations that never rise to our conscious attention. So like, for example, my stomach is currently at some pH. I couldn't tell you exactly what. I could estimate since I haven't eaten in a while. But via sensory information from chemoreceptors in my stomach, my brain sure knows what the pH is. I just don't because it doesn't rise to my conscious attention. So that's an example of a visceral sense. So let's add cell bodies of, and then we'll say cell bodies of, I'll just erase the central canal. Okay, and then in the posterior, or excuse me, the anterior gray horn, see their stupid mislabeling is screwing me up. So we established that pink is our color for efferent. So we have cell bodies of motor neurons for the somatic nervous system, and then a little bit above for the autonomic or visceral motor neurons. So let's label those. So get rid of these commissures here. Those colors aren't quite as different as I would like them to be, so well, that's even worse. I'm just going to go ahead and assign this a reddish color. I think that will be better. Yeah, there we go. Okay. So these are the cell bodies of somatic motor neurons, and these are the cell bodies of visceral / autonomic motor neurons. And so just to give you a quick little reminder, connect the, identification lab stuff with lecture stuff, somatic motor neurons are going to be innervating skeletal muscle. And that's the effector of the somatic nervous system. And then autonomic or visceral motor neurons are the other effectors. So smooth and cardiac muscle, glands, and adipose tissue. So effectors are the things that the motor neurons control, and there is this very stark division between visceral and somatic, and the autonomic nervous system and stuff you have voluntary control over. So there's also a lateral gray horn on each side, and these are actually associated with the thorax, so it's a little bit weird that they're so visible up here. But this is the lower C spine, so this is C6 and C7, so I guess it's not inconceivable that you would see some gray hornage here. It's just a little bit exaggerated in this model. And that is associated with sympathetic nervous system stuff that we'll get to later, so I'm not going to overemphasize it here. Okay, So let's have a peep at some other stuff. I'm not going to address these tracts until we get to the appropriate lecture stage. But I do want to point out that we've got our three mothers here. So another error that I'm noticing is that these two words should have a space between them. So dura space mater, which means tough mother. Mater means mother in Latin. Arachnoid mother means spider-like mother. And pia, again, there should be a space here, means soft mother. So the fat-filled space outside of the dura mater, between the dura mater and the bone of the vertebra, is the epidural space. Between the dura mater and the arachnoid mater is the subdural space. Underneath the arachnoid mater, and I'm just going to go ahead and write it in here, is the subarachnoid space. And that's filled with cerebrospinal fluid. And then the pia mater actually touches the surface of the nervous tissue itself. So the subdural space is really a potential space. So under ordinary conditions, there's not this much of a gap. The two layers, the dura mater and the top of the arachnoid mater, are touching each other, but they're not like stuck to each other. The subarachnoid space should be filled with cerebrospinal fluid to cushion and nourish and provide circulation and waste removal for the spinal cord, as well as the CSF that's going down the central canal. If you are going to get epidural anesthesia, which is common for anesthesia-aided labor and delivery of a baby, they usually just call it an epidural. But in that case, the location where the cannula goes, and I'm saying cannula on purpose, it's not a needle, is in the epidural space. So a cannula has a blunt end, and the anesthetic flows out through the hole in the top. And the reason for that is that we don't want to accidentally stab through and have the needle enter the spinal cord. That would be really bad. So instead, we can avoid that by having a blunted cannula positioned there as well. So in that way, the anesthetic will diffuse into the spinal cord and deaden the sensory information so that it doesn't hurt so much. And then in the epidural space, there's also a lot of arteries and veins. So the anesthesia is also able to diffuse into those vessels. So that's how an epidural works. Okay, and that is that. So I'm going to end this video and go find some models for some other stuff I want to talk about. Thank you for your attention, and I'm going to go post this right now.